Good day, it's Dr. Will, CBMD PhD. Hey, Sunday, January 3rd, 2016. It is 9:57 a.m. Eastern Standard Time. Continuation. Organisms whose cells have a nucleus are called eukaryotes, from the Greek words eu meaning well or truly, and a kernel or nucleus. Organisms whose cells do not have a nucleus are called prokaryotes, from pro meaning before. The terms bacterium and prokaryotes are often used interchangeably, although we shall see that the category of prokaryotes also includes another class of cells, the archaea, singular archean, which are so remotely related to bacteria that they are given a separate name. Prokaryotes are typically spherical, rod-shaped or corkscrew-shaped, and small, just a few micrometers long. Although there are some giant species as much as 100 times longer than this, they often have a tough protective coat called the cell wall surrounding the plasma membrane, which encloses a single compartment containing the cytoplasm and the DNA. In the electron microscope, the cell interior typically appears as a matrix of varying texture without any obvious organized internal structure. The cells reproduce quickly by dividing in two. Under optimum conditions, when food is plentiful, a prokaryotic cell can duplicate itself in as little as 20 minutes. In 11 hours, by repeated divisions, a single prokaryote can give rise to more than 8 billion progeny, which exceeds the total number of humans presently on Earth. Thanks to their large number, rapid growth rates, and ability to exchange bits of genetic material by a process akin to sex, populations of prokaryotic cells can evolve fast, rapidly acquiring the ability to use a new food source or to resist being killed by a new antibiotic. Prokaryotes are the most diverse of cells. Most prokaryotes live as single-cell organisms, although some join together to form chains, clusters, or other organized multicellular structures. In shape and structure, prokaryotes may seem simple and limited, but in terms of chemistry they are the most diverse and inventive class of cells. These creatures exploit an enormous range of habitats, from hot puddles of volcanic mud to the interiors of other living cells, and they vastly outnumber other living organisms on Earth. Some aerobic, using oxygen to oxidize food molecules, some are strictly anaerobic and are killed by the slightest exposure to oxygen. Virtually any organic material from wood to petroleum can be used as food by one sort of bacterium or another. Even more remarkably, some prokaryotes can live entirely on inorganic substances. They can get their carbon from CO2 in the atmosphere, their nitrogen from atmospheric N2, and their oxygen, hydrogen, sulfur, and phosphorus from air, water, and inorganic minerals. Some of these prokaryotic cells, like plant cells, perform photosynthesis, getting energy from sunlight. Others derive energy from the chemical reactivity of inorganic substances in the environment. In either case, such prokaryotes play a unique and fundamental part in the economy of life on Earth. Other living things depend on organic compounds that these cells generate from inorganic materials. Plants too can capture energy from sunlight and carbon from atmospheric CO2, but plants unaided by bacteria cannot capture N2 from the atmosphere, and in a sense even plants depend on bacteria for photosynthesis. It is almost certain that the organelles in the plant cell that perform photosynthesis, the chloroplasts, have evolved from photosynthetic bacteria that found a home inside the plant cell's cytoplasm. The world of prokaryotes is divided into two domains, bacteria and archaea. Traditionally, all prokaryotes have been classified together in one large group, but molecular studies reveal that there is a gulf within the class of prokaryotes, dividing it into two distinct domains called bacteria, sometimes eubacteria. In the archaea, remarkably at a molecular level, the members of these two domains differ as much from one another as either does from the eukaryotes. Most of the prokaryotes familiar from everyday life, the species that live in the soil or make us ill, are bacteria. Archaea are not only found in these habitats but also in environments hostile in most other cells. There are species that live in concentrated brine and hot acid volcanic springs, in the airless depths of marine sediments, in the sludge of sewage treatment plants, and pools beneath the frozen surface of Antarctica, and in the acidic oxygen-free environment of a cow's stomach, where they break down cellulose and generate methane gas. Many of these environments resemble the harsh conditions that must have existed on primitive Earth, where living things first evolved before the atmosphere became rich in oxygen. The eukaryotic cell. Eukaryotic cells in general are bigger and more elaborate than bacteria and archaea. Some live independent lives as single-cell organisms, such as amoeba and yeast. Others live in multicellular assemblies. All of the more complex multicellular organisms, including plants, animals, and fungi, are formed from eukaryotic cells. By definition, all eukaryotic cells have a nucleus, but possession of a nucleus goes hand in hand with possession of a variety of other organelles, subcellular structures that perform specialized functions. Most of these are likewise common to all eukaryotic organisms. Mitochondria generate usable energy from food to power the cell. Mitochondria are present in essentially all eukaryotic cells, and they are among the most conspicuous organelles in the cytoplasm. These organelles have a very distinct structure when seen with an electron microscope. Each mitochondrion appears sausage or worm-shaped from 1 to many micrometers long, and each is enclosed in 2 separate membranes. The inner membrane is formed into folds that project into the interior of the mitochondria. Mitochondria contain their own DNA reproduced by dividing in 2. Because mitochondria resemble bacteria in so many ways, they are thought to derive from bacteria that were engulfed by some ancestor of present-day eukaryotic cells. This evidently created a symbiotic relationship in which the host eukaryote and the engulfed bacterium helped one another to survive and reproduce. Observation of the microscope by itself gives little indication of what mitochondria do. Their function was discovered by breaking open cells and then spinning the soup of cell fragments in a centrifuge. This separates the organelles according to their size, shape, and density. Purified mitochondria were then tested to see what chemical processes they could perform. This revealed that mitochondria are generations of chemical energy for the cell. They harness the energy from the oxidation of food molecules such as sugars to produce adenosine triphosphate or ATP, the basic chemical fuel that powers most of the cell's activities. Because the mitochondria consumes oxygen and releases carbon dioxide in the course of this activity, the entire process is called cellular respiration, essentially breathing on a cellular level. Without mitochondria, animals, fungi, and plants would be unable to use oxygen to extract the maximum amount of energy from the food molecules that nourish them. Oxygen would be a poison for them rather than an essential requirement. That is, they would be anaerobic. Many prokaryotes are anaerobic, and there are even a few anaerobic eukaryotes, such as the intestine parasite Giardia, that lack mitochondria and live only in environments that are low in oxygen. Greek alphabet: Alpha, Beta, Gamma, Delta, Epsilon, Zeta, Eta, Theta, Iota, Kappa, Lambda, Mu, Nu, Xi, Omicron, Pi, Rho, Sigma, Tau, Upsilon, Phi, Chi, Psi, Omega,